Hello viewers, welcome to my channel and this bioteaching. In the previous class, I already discussed about glycolysis and today the topic is Krebs cycle. So let's start the video. Krebs cycle. Krebs cycle, the central metabolic pathway are the major source of energy in living organisms. The Krebs cycle, also known as citric acid cycle or the tricarboxylic acid cycle, which is called in brief TCA cycle also. This cycle involves oxidation of acetyl-CoA to carbon dioxide and H2O. This is the main function. Final oxidative pathway for carbohydrates, fats, amino acids. Oxidative decarboxylation of pyruvate to acetyl-CoA by pyruvate dehydrogenase complex, which is called PDH complex. This step connecting link between glycolysis and TCA cycle. That means this is the middle stage. There is no direct participation of oxygen. This is very important fact and operates only under aerobic conditions. These points I will discuss broadly. Here you can see the link reaction between glycolysis and Krebs cycle. Here what happened? Conversion of pyruvate to acetyl-CoA. Pyruvate transported across the mitochondrial inner membrane, that means in matrix, and enzyme pyruvate dehydrogenase complex splits pyruvic acid into a carbon dioxide and 2 carbon of acetyl-CoA, which enter into the Krebs cycle. In this picture you can see clearly. Here you can see that glycolysis, glucose converted to pyruvate, then pyruvate converted to acetyl-CoA, where pyruvate oxidation occur and enter into the citric acid cycle or Krebs cycle where it occurs in the mitochondrial matrix. And this in the citric acid cycle, which produce FADH2 NADH, which enter into the oxidative phosphorylation, that is electron transport chain system. It is also called oxidative phosphorylation. Occurrence, where occur this Krebs cycle? The Krebs cycle takes place in the mitochondria of the cell. The inner membrane of mitochondria surrounds the mitochondrial matrix where the process of Krebs cycle, this is mitochondria and this is the matrix of the mitochondria where takes place. First of all, we have to know what is its purpose. Each and every cycle have purpose, so we have to clear about the purpose of each cycle. Then what is the purpose of Krebs cycle? Conversion of acetyl-CoA to carbon dioxide. This is the main purpose. Conversion of acetyl-CoA to carbon dioxide generates reducing equivalents, that is NADH plus, H plus, FADH2 and GTP to be oxidized to be in the respiratory chain to generate ATP. Main function is ATP generate. Coming to the whole cycle, first step, step 1, formation of citrate. Acetyl-CoA, which is 2 carbon molecule, acetyl-CoA joined with oxaloacetate, which also called oxaloacetic acid, which is 4 carbon molecules, to form citrate. This is 2 carbon and this is 4 carbon, then form here 6 carbon molecule of citrate or citric acid. Step 2, here formation of isocitrate. Citrate is converted to isocitrate, which is isomer of citrate. Here you can see that this is citrate or citric acid, which converted to cis-aconitic acid, then converted isocitric acid. Here which occur? Isomerization occur. Step 3, oxidation of isocitrate and formation of carbohydrate. How? Isocitrate is oxidized to alpha ketoglutarate, which is 5 carbon molecule, which results in the release of carbon and carbon dioxide. 1 NADH molecule is formed. Here in this picture you can see clearly isocitrate acid is converted to alpha ketoglutarate. Here manganese or magnesium also require and NADH molecule is formed and release of carbon dioxide and 1 NADH molecule. Then step 4, oxidation of alpha ketoglutarate and formation of carbon dioxide. Alpha ketoglutarate, that means alpha ketogluteric acid, which is 4 carbon molecule. This binds to coenzyme A and forming succinyl CoA. A second molecule of NADH is produced alongside a second molecule of carbon dioxide. Step 4, phosphorylation. Succinyl CoA is then converted to succinate, which is 4 carbon molecule. This 4 you can see 1 2 3 4 carbon molecule and 1 GTP molecule is produced. Here the enzyme involved succinyl thiokinase. Step 6, oxidation of succinate. Succinate is converted into fumarate or fumaric acid, which is 4 carbon molecule and a molecule of FADH2 is produced. Hydration of fumarate. Fumarate is converted to malate, which is 4 carbon molecule. Then oxidation of L-malate to regenerate oxaloacetate. Step 8 here. Malate is then converted to oxaloacetate. Here also it is 4 carbon molecule, and the third molecule of NADH is also produced. Here the enzyme involved malic dehydrogenase. Here you can see the whole pathway in cyclic manner. Oxaloacetic acid, you can observe very clearly step by step that oxaloacetic acid and acetyl-CoA together converted to citric acid, which is 6 carbon molecule, and then citric acid converted to cis-aconitic acid with the help of the enzyme aconitase. Cis-aconitic acid then converted to isocitric acid, which is also 6 carbon molecule with the help of the enzyme aconitase. Isocitric acid here which happen isomerization occur here, and then isocitric acid converted to alpha ketoglutaric acid, which is 5 carbon molecule with the help of isocitrate dehydrogenase. Then alpha ketoglutaric acid converted to succinyl-CoA. Here the enzyme involves succinate dehydrogenase. Here NADH produced. This is 4 carbon molecule. Then succinyl coenzyme converted into succinic acid with the help of the enzyme succinate thiokinase. Here direct ATP is produced. This step is called substrate level phosphorylation due to direct ATP formation. Then succinic acid converted to fumaric acid with the help of the enzyme succinate dehydrogenase. Here FADH2 produced. Then fumaric acid converted to malic acid. Here the enzyme involved fumarate, and here 1 molecule of water released. And then malic acid converted to oxaloacetic acid with the help of the enzyme malate dehydrogenase. Here also NADH produced, and then here NADH, and 3 NADH molecule produced and 1 ATP molecule here produced. Then oxaloacetic acid converted into citric acid. In this way the whole cycle operates in the matrix of the mitochondria. The enzymes are very important. You have to remember the enzyme's name and each step of this cycle. Then practice and you can learn it very easily. Now we have to know net output means what is produced in the ultimate products. Each cycle produces 2 molecules of carbon dioxide. In the previous slide you have seen that 2 molecules of carbon dioxide here and here, and here 2 molecules of carbon dioxide are produced. 3 molecules of NADH, 3 molecules of NADH, here NADH, and here NADH. 3 molecules of NADH are produced and 3 hydrogen ions, 1 molecule of FADH2, 1 molecule of FADH2 here you can see, and 1 molecule of GTP. Here 1 molecule of GTP, like similar to ATP. This is the whole output of the Krebs cycle. Energetics in this pathway: oxidation of 3 NADH of ETC with oxidative phosphorylation results in the synthesis of 9 ATP. APD leads to the formation of 2 ATP, 1 substrate level phosphorylation. Thus, a total 12 ATP are produced from 1 acetyl CoA. Number of ATP generated oxidation 3 NADH means 9 because 1 NADH equal to 2.5 ATP. We can suppose whole in round figure 3. That's why the here ATP production is 9. Then number of ATP generated by oxidation of APD is 2 and number of ATP generated from GTP 1. Then total 12 ATP are produced in one molecule of acetyl CoA. Isocitrate dehydrogenase is activated by citrate synthase is inhibited by ATP, NADH, acetyl CoA and succinyl CoA. Isocitrate dehydrogenase is activated by ADP and inhibited by succinyl CoA and NADH. Alpha ketoglutarate dehydrogenase is inhibited by succinyl CoA and NADH. Availability of ADP is very important to proceed cycle. Then coming to the significance of Krebs cycle, acetyl CoA, ATP generation, integration of major metabolic pathway, fat is burned on the wick of carbohydrate, excess carbohydrate are converted as neutral fat. No net synthesis of carbohydrates from fat, carboskeleton of amino acids finally enter the TCA cycle. These are the significance of whole Krebs cycle. This is the final slide, and you read very carefully those slides and pause the video and write down if you require these notes. Okay, I am ending here. If you are helpful from this video, then please like, share and comment, and if you are new in my channel, then subscribe my channel and click the bell icon to get new notification. Thank you.